Listen me, and thank you the organizers of this meeting, Dr. Foa, Dr. Wilson, and everyone. Um, um, we all are Latin Americans in almost Latin Americans in this meeting, so you will have no problem to understand my perfect Spanglish at this time. So apologize for that. In the next 10 minutes, we will talk about cellular biology of B-cell lymphomas. My name is Jorge Vela from Mexico City, by the way. So, naive B cells coming from bone marrow enter the germinal centers or the primary B cell follicle using the in the T cell zone the high endothelial venule. Once the B cell comes to the secondary lymphoid organs, such as lymph nodes or maybe spleen, uh, we have three checkpoints in this region. The first checkpoint to enhance the BCR affinity occurs firstly in the subcapsular region by presentation of antigen by macrophages of the subcapsular zone and dendritic cells, and also in soluble form. So B cell receive the antigen presented by these cells. After that, B cells exit the primary follicle and encounter another checkpoint uh, conferring by CD4 T cells. As you can see, a second checkpoint and a second antigen presentation, in this case by CD4 T cells. And after that, the B lymphocyte is ready to get the secondary follicle. As you know, as all of you know, secondary cell follicle is composed of four zones: the marginal zone, in the periphery, the mantle zone, and the germinal center, composed by two zones, the dark zone and the light zone. In the dark zone, B cells are highly proliferating, the so-called centroblast, and in the light zone, the centrocytes are proliferating, but in order to enhance the BCR affinity, a third checkpoint occurs. It is the presentation of antigen by follicular dendritic cells and follicular T helper cells. So in order to increase the BCR affinity, the first checkpoints in the subcapsular zone, the second one by CD4 T cells, and the third one by follicular dendritic cells and TH cells. This process, this direction of the immature B cells from the dark zone to the light zone is regulated by some genes, some important genes. For example, these three genes, PAX5, BCL6, and the other one regulates the transit between dark zone and light zone. And other genes, for example, NF-kappa B, IRF4, are regulating especially the B cells in the light zone. In order to exit the follicle, the B cells, these hens have to switch off, for example, NF-kappa B and IRF4, and in this way, B cells transform in memory B cells or immunoglobulin secretory plasma cells. So if we know this phenomenon, we can explain the origin of non-Hodgkin lymphoma. For example, Burkitt's lymphoma originates in the dark zone, mainly. Follicular lymphoma and germinal center diffuse large B-cell lymphoma originates in the light zone. And the so-called ABC diffuse large B-cell lymphoma originates in the plasmablast phase of the differentiation. And lymphomagenesis can be explained, for example, in the case of Burkitt's lymphoma, by the aberrant expression of MYC, a product of the three translocations that we know, and the signaling, the intracellular signaling pathways, mainly by PI3K kinase activation. In the case of follicular lymphoma, we have two important biological phenomena: the activation of BCL2 blocking apoptosis, and the aberrant expression and inhibition of three molecules of the epigenetic methyltransferase and epigenetic phenomenon that are blocked. In the case of diffuse large B-cell lymphoma, the so-called germinal central B-cell lymphoma is also explained by the inhibition of some methyltransferase at the level of epigenetics. And in the case of ABC diffuse large B-cell lymphoma, by constitutive activation of NF-kappa B. So in terms of treatment, it is very easy to use drugs that normally act at the epigenetic level in diffuse large B-cell lymphoma germinal center origin, and inhibitors of NF-kappa B in the case of ABC diffuse large B-cell lymphoma. And the microenvironment is very important, not only to explain the proliferation of malignant cells, but also microenvironment confers multi-drug resistance. We have three patterns of microenvironment in lymphomas in general. The first pattern, re-education, is observed as an example in follicular lymphoma, in which we have a lot of tumoral cells dependent of the microenvironment. So the contact between the stromal cells and tumoral cells produce a dependence of the tumoral cells in the microenvironment. The second pattern is explained, for example, in Hodgkin's lymphoma. This type of patterns, you can see a few tumoral cells and a lot of reactive cells that secretes interleukins, growth factors, chemokines, and some kind of soluble factors that made the tumoral cell immortal. So this is the case in Hodgkin's lymphoma. And the third pattern, in which the tumor is independent, has is growing independently of the microenvironment, and for example in Burkitt's lymphoma, in which the presence of MYC by the product of the translocation made this tumor independent of the microenvironment. The other point is the drug resistance. In lymphomas, we have two types of drug resistance regulated by microenvironment. The first one is the soluble resistance made by the secretion of tumor-associated stroma, the secretion of growth factors, chemokines, and chemokine receptors that make the cell resistant to drugs. The second one is the so-called cell adhesion resistance, that is regulated by integrins that contact the synapses between the tumoral cells and the stromal cells, make these tumoral cells resistant to any kind of chemotherapy and even radiotherapy. Another important point is that more than 60% of lymphomas, such as in the case of diffuse large B-cell lymphoma, in this kind of lymphoma, there is a lack of major histocompatibility complex in the surface of the tumoral cells. And as you know, this molecule is very important in the activation of cytotoxic T cells. So in non-Hodgkin's lymphoma, for example, diffuse large B-cell lymphoma, this is a mechanism of immune surveillance blockage in this kind of lymphoma. And also 60% of the lymphoma patients has aberrant expression of CD58, a molecule very important in the activation of NK cells. Another important point in microenvironment is the presence of some soluble proteins or some components proteins that such as matrix metalloproteinase 2 and 9. As you can see in this work, osteopontin, IL-6, metalloproteinase 2 and 9 are increased in patients with diffuse large B-cell lymphoma in comparison with normal controls. These findings can explain the adhesion, the drug resistance, the proliferation, and the resistance to chemotherapy. Another important factor, p53. As you can know, p53 is our protein, the most important protein that protects from cancer. p53 has a regulator, MDM2. In order to function, p53 has to blockade MDM2 and inducing apoptosis, DNA repair, governing cell cycle, etc. But in the case with mutations of, for example, 17p deletion in some cancers, MDM2 is blocking TP53, and this results in inflammation, proliferation, angiogenesis, cancer progression, tumorigenesis, and also metastasis. p53 has 11 exons. The last ones, 6, 8, 9, and 10, regulates some enzymes processes, acetylation, methylation, that this enzymatic process blockages the repressor of p53, and this is the way why the p53 function. And my last slide is talking about mantle cell lymphoma for its importance in treatment. As you can see, the same phenomenon. Lymphoma-associated macrophages, stromal cells secrete chemokine receptors, growth factors, all of these to get in touch the cancer cell and the stromal cell, conferring resistance, proliferation, increased angiogenesis, etc. And BCR, B-cell antigen, will be touched in the next talk more extensively. So in conclusion, hematologists must know the normal process of B-cell development. Distinct types of germinal center-derived lymphomas originates from normal counterpart cells that are blocked at different stages of maturation. Tumor microenvironment plays a crucial role not only into the genesis of lymphomas, but in the mechanisms of multi-drug resistance in an immune escape. And p53 alteration is a frequent finding in all kinds of cancers, but also in lymphoma cells, and confers poor prognosis. Thank you very much for your attention, and this talk is open for discussion. Thank you.
So I think we have time for one or two questions. So I just wanted to say that I thought this was a really excellent overview, and I think that it is critical to understand the normal biology because that is what is going to instruct us on what the actual targets are, and also to be able to understand that when you're using targeted therapy, the treatment of one type of tumor is going to be very different from another. We're going to hear the second talk is going to be on targeting the B-cell receptor signaling, and as we heard from your talk, that is not turned on, at least not in normal cells from which Burkitt comes from. So you would not expect a drug like that to actually work, but it is turned on in the post-germinal center type tumors, and that would include, of course, ABC large cells. So I think that if you really want to understand how all the tumorigenesis interfaces, you really do need to understand this basic underpinnings of these cells.
I got a quick question because you brought up I thought just fascinating between the microenvironment, but the epigenetic approaches and some of the mutations is some of the things we're seeing, because we know that some epigenetic changes is reasons of resistance and all. Are some of those mutations within the proteins that then cause the increase, methylation or signaling that way? So is the epigenetic changes due to mutations in these cells, or is it more the cellular process that's occurring?
Yes, I agree. The epigenetic phenomena are very important in all kind of cancers, and we are not taking into account in terms of treatment. So maybe the combination of the normal drugs we are giving in lymphomas combined with drugs that act into epigenetic phenomena could increase the degree of response of our patients. I think that there is one important paper showing that epigenetics phenomena like inhibition of these methyltransferases are related to drug resistance also. So this is very exciting field to research even.
So, I mean, I think your question was, are these changes driven by actually mu mutations? Well, the answer is yes. There's a whole host of mutations that are seen. However, a lot of these can be turned on functionally as well. So it's very much of a mixed picture.
So it's going to be, you're going to really have to approach it kind of individually again.
That's correct.
What's happening within the microenvironment at that time.
Right.
And for example, the microenvironment is very important. We are doing this in monoclonal gammopathies, for example. Lenalidomide, even thalidomide, bortezomib inhibits the contact, the synapses between tumoral cells and stromal cells, and this overcomes the drug resistance observed in these patients. Is the same in lymphomas and many other cancers. Thank you.
Oh, yes.
Yes, I'd like to make a comment on trying to integrate this in our clinical practice. Regarding diffuse large B-cell lymphoma, we still don't have a very good method to distinguish between germinal center and ABC type. And thinking about these new drugs, I think it's very important because in the future, these might be two different diseases, and we need to treat them first line differently. So I think all this information regarding diffuse large B-cell lymphoma, we're still not ready to incorporate in our clinical practice.
So that's a very good point, and many people have raised this. Of course, there's the Hans method. However, the Hans method isn't that accurate. We now have a nanostring assay that is actually going to be going to the FDA, and so there will soon be a very robust assay using nanostring. And as long as your centers or a center has a nanostring system, it is actually based on 19 genes, so the cost is actually going to be relatively low.